I'm going to teach biochemistry, which is module 1. First, we're going to study about the atomic structure. Atom. What is an atom? Atom is the smallest unit that retain chemical and physical properties of a particular element. Atom consists of 3 parts: proton, neutron, and electrons. Protons carry positive charge, neutrons carry no charge, like they're neutral, they doesn't have any charge, whereas electrons have negative charge. Protons and neutrons are located in nucleus. Number of protons present in an atom will define elemental identity. Atomic number is always equal to number of protons. For example, carbon 6. It means carbon has 6 protons and 6 electrons, whereas number of neutrons can vary. What is mass number? Mass number is the number of protons plus number of neutrons. Here is an example of atomic symbol. Potassium, here is an element we are using. It has 19 is its atomic number and 39 is its mass number. It means potassium has number of proton is equal to 19 and the mass number of proton, which is proton plus neutron, is equal to 39. So if we want to find number of neutrons, we can simply subtract mass number minus atomic number. So 39 minus 19, it gives us 20, which is number of neutrons. Now we're going to discuss about isotopes and radioisotopes. Forms of element that differ in its number of neutron is called isotope. Isotopes have same behavior in chemical reaction because its number of neutron which varies in isotopes. The isotope have same number of protons and electrons which are responsible for the chemical behavior, whereas radioisotope can be defined as a radioactive isotope of an element. For example, carbon have 3 forms: C12, C13, C14. In each carbon here, the number of electron and proton is 6, whereas number of neutron varies. C12 has 6 neutrons, C13 has 7 neutrons, and C14 has 8 neutrons. C14 is very unstable in nature. Radioactive isotopes are used in numerous medical applications, and also they are used in instrument to elucidate the structure of unknown compounds. Radioactive isotopes, they gives radioactive signals when they decay, so they can be easily detectable in cell. Scientist Melvin Calvin uses carbon 14 labeled molecule and he discovered the process of photosynthesis. Most of the biological compounds contain carbon and hydrogen, so C14 and H3 used as tracers. They are also used in new field of nuclear medicine to help various diagnoses of diseases and then their treatments. Okay, now we will study chemical bonds. Atoms form stable attractions to one another called chemical bonds. Chemical bonds are further of 4 types: ionic, covalent, and the other 2 are types of forces, which are intermolecular forces and van der Waals forces. First, ionic bonds. Ionic bonds form between atoms that have lost or gained electrons to become charged. Atom that have lost or gained electrons are called ions. Atoms which has lost or gained are called ions. Ions can be positively charged now or negatively charged. The positively charged ion called cation, negatively charged ion called anion. You can remember it by CP. Cations are positively charged or not applicable. That means anions are negatively charged. That's the way how I remember it. Now, the ions are strongly attracted to water. Hence, they easily dissociate and dissolve in water. Because of the strong attractions, they easily dissociate and dissolve in water and form hydrate ions. Okay. Covalent bonds. How these bonds are formed? When atoms share one or more pair of valence electron. The electrons present in outermost shell is called valence electron. Then the covalent bonds formed. Formation of H2 is the simplest example of covalent bond. Here I am just writing CP for covalent bond. So hydrogen has 1 and 1 electron and then they share and then it forms a covalent bond. Number of covalent bonds is equal to number of additional electrons needed to fill its valence shell. Carbon has 4 electrons in the valence shell. That means outer shell carbon has 4 electrons. Therefore, bonds formed with 4 hydrogen atoms to form compound called methane. So carbon shares all 4 electrons with 1 electron of hydrogen and form CH4. Polar molecules. All covalent bonds involve sharing of valence electron. They differ widely in degree of sharing takes place. The more electronegative atom, the more strong it attracts electron. The more the electronegative atom, more strongly it attracts the electron. Electronegativity, I'm just writing En here, is influenced by atomic number. And the second factor which determines the electronegativity is distance between valence electrons and nucleus of the atom. What is the distance between electron and nucleus? As the distance decreases, the electronegativity increases. So if the electron is more close to nucleus, the electronegativity is gonna be higher. Here is the example. Oxygen has very high electronegativity because the additional electron can occupy valence orbital which are very close to the nucleus of oxygen. Since the distance is less, hence the electronegativity of the oxygen is high. Unequal sharing of electron, I'm just using E negative for electron here. Unequal sharing of electron between two atoms with different electronegativity result in polar covalent bonds. Atoms that attract the valence electron more strongly carry partial negative charge and the other one will carry partial positive charge. Because atoms carry partial charges, the whole molecule have non-uniform charge distribution. This is called polarity of the molecule. Polar molecules attract and align themselves to another polar molecules and tend to be soluble in water. The oils are non-polar molecules, hence they are not soluble in water. Methanol has partial positive charge. Negative charge of the oxygen in the water attracts ends of the methanol which has positive charge. So like methanol end has positive charge and the oxygen has negative charge. They attract each other and hence it is dissolvable in water. Here is the example showing methanol pulled toward water. Intermolecular forces are known as van der Waals forces. They are very weak forces of attraction between two molecules. They are extremely important because they influence solubility, melting point, and brittleness. Strength of these forces are dependent on size and polarity of molecule. These two factors will determine the strength of the forces. You can say SSP, that's how you can remember. Strength will be determined by size and polarity. Hydrogen bonds. A hydrogen atom that is covalently bonded to strongly electronegative atom in one molecule, such as oxygen or nitrogen, can attracted to strongly electronegative atom in different molecule. The resulting attractive forces are called hydrogen bond. Bonds are strong. Hydrogen bonds are strongest and significant form of van der Waals forces. Individual hydrogen bonds are weak as compared to ionic and covalent bonds. They are weaker, but they are significantly strong when they occur in large numbers. The hydrogen bonds that exist between water molecules are responsible for many properties that makes water a unique molecule. For example, heat capacity, high melting and boiling point, cohesion, adhesion, surface tension. Trees depend on cohesion. Adhesion of water to xylem cell wall of plant helps to contract the downward pull of gravity. That's how water will move up against the gravity because of adhesion. Other van der Waals forces. Size and shape influence the total strength of these forces. You can say SSV, size and shape. Methane, CH4, is a gas at room temperature, whereas octane, which is a large molecule, it is liquid at room temperature. Now we will study about chemical reactions. There are four major chemical reactions: dehydration reaction, which are also called as condensation reaction, hydrolysis reaction, neutralization reaction, and redox reaction. Dehydration. It is the removal of OH or hydrogen bonds from two reactant molecules. When these bonds are removed, it means dehydration. Here is an example. Here there is a loss of water to form the product. OH and H bonds form water molecule, while other two reactant molecules join together. When reaction takes place, dehydration reaction takes place, water just formed as a byproduct. Used by cell to join, dehydration reactions are used by cell to join smaller molecules and assemble extremely large molecules such as complex carbohydrates and proteins. Now hydrolysis. Hydrolysis is the reverse of dehydration. In dehydration, there is a removal of water. In hydrolysis, there is addition of water. You can see here. Neutralization reaction occurs between acid and bases. To produce salts. Water produced as reactant. Here is an example. Hydrochloride plus sodium hydroxide react. The product is sodium chloride, which is table salt, and water. Redox reactions. Electron lost from one atom and gained by other. Oxidation means loss of electron and reduction means gain of electron. Redox reaction. Oxidizing agent is a molecule that is being reduced. In redox reaction, oxidizing agent is the molecule which is getting reduced, and reducing agent is the molecule which is getting oxidized. Here is the example. This thing Xe is losing electron and becoming X here, whereas Y gaining electron, it's reduction. CH4 methane reacts with oxygen and it is making carbon dioxide. Now this is oxidation, whereas we can say O2 is reducing agent in this case. Similarly, oxygen is becoming water here by reduction I mean, or you can say electron gaining. Here methane will be oxidizing agent. Water is a universal solvent. Water molecules is special because of their shape, size, polar structure, and ability to associate with each other through hydrogen bond. These properties are responsible for universal nature of water. In liquid, each water molecule forms 3.4 hydrogen bonds with neighboring water molecules. Binding forms arrangement called water lattice. Water lattice is unique feature of water. That's the reason why ice never sinks in water. In liquid water, hydrogen bonds that hold lattice together constantly break and reform, allowing water molecules to slip past one another and reform the lattice in new position. This gives water its fluid property. Each water molecule in ice forms 4 hydrogen bonds with neighboring water molecules. The rigid ice water lattice space the water molecules further apart than they are in the liquid water lattice. Because of the greater spacing, water has a property of being 10% less dense than its solid state than in its liquid state. So now if the water is 10% less dense in its solid state than the liquid state, that's the reason why ice floats on the water. As a result of its stabilizing hydrogen bond lattice, water has a high specific heat capacity. What is specific heat capacity? Amount of heat, or you can say thermal energy, that is required to increase the temperature of a given quantity of water by 1 degree of Celsius is called specific heat. High boiling point of water ensures that it is liquid state from 0 degrees Celsius to 100 degrees Celsius. Without hydrogen bond, water would boil at 81 degrees Celsius. And then if the water is boiling at 81 degrees Celsius, most of the water is gonna be gas, and then there will be no drinkable water, no swimming, no other leisure activities possible. Hydrogen bond lattice of water result in water molecules staying close together. This property is called cohesion. Surface tension is the measure of how difficult it is to stretch out or break the surface of liquid. Because of the surface tension, insects can walk on the surface of water. Water molecules can form hydrogen bonds with other polar molecules. This property is called adhesion. With each other is cohesion, and with other is called adhesion. And I would like to make one more point here, surface tension. So water molecule can form bonds with all three other hydrogen except the part which is exposing to air. That's why there is a very strong forces of attraction at the surface of water, which helps insects to walk on the surface of water. Cohesion helps water molecules to stick together. Adhesion helps water molecules to stick to the walls as water is transported. Aqueous solution. It can be hydrophilic, hydrophobic. Philic means lover. Phobic means scared. Molecules which love water are easily dissolvable in water, called hydrophilic molecules. The hydrophobic molecules are scared of water, that's why they are not soluble in water. Ionization and pH. Most critical property of water aside from hydrogen bonding is the ability to dissociate into ions. Acids and bases. When the concentration of hydronium ion H3 positive in a solution is greater than the concentration of hydroxide ions, the solution has properties of an acid. If H3 positive ions are greater than OH negative, it is an acid, which is, we know the properties of acid are sour taste, ability to conduct electricity, turns blue litmus paper red. When the concentration of OH negative ion in a solution is greater than the concentration of H3 positive ion, the solution is basic in nature. If H3O positive is less than OH negative, properties slippery, bitter taste, ability to conduct electricity, and turns red litmus paper blue. So bases turn red litmus paper blue and acid turns blue litmus paper red. In pure water, there is a balance in concentration of H3O positive ions and OH negative ions. So the pure water is neutral. Solution with pH less than 7 is acidic. If it is greater than 7, it's basic. If it is equal to 7, it's new. pH within the cell and outside cell. Both are important for proper functioning of cell. Change of even 0.1 or 0.01% in pH level can drastically affect biological reaction. pH of an ocean is 8. Burning of fossil fuels leads to more CO2 production, and then hence CO2 is gonna enter in oceans, which is gonna be making more carbonic acid, and then oceans will be more acidic, hard for aquatic animals to survive. Now we're going to discuss about strong and weak acid bases. Acid and bases can be classified as strong or weak. The strength of an acid or base depends upon the degree to which it ionizes when dissolved in water. A strong acid and base completely dissociate in water. The strong acid is HCl and strong base is NaOH, which release OH negative ions and increase the hydroxide ion concentration of water in which they are dissolved. Weak acid bases only partially ionize the water. Here is an example. CH3COOH is a weak acid. It's gonna react with water and form CH3CO negative plus H3 positive O. Neutralization reactions and buffers. When an acid and base reacts, product is salt and water. HCl, acid, base, NaOH, water, and salt is formed. Living organisms have control over internal pH by using buffers. Buffer is a chemical substance that compensates for pH changes in a solution by either accepting or donating H positive ions. When a biological reaction releases H positive ions, excess buffer combine with H positive ions, so they can no longer be free in solution. Conversely, if the concentration of H positive ion decreases, the buffer releases H positive ion. So if it increases, the buffer absorbs H positive ion. If the H positive ion concentration decreases, it goes other way around, and then the buffer releases H positive ions. Since the weak acids and bases dissociate in water, most buffers are either weak acids or weak bases, or combination of both. In humans and many other animals, buffering system is based on carbonic acid, H2CO3, which helps to maintain proper blood pH level. Here is a reaction. H2CO3 dissociate and releases H positive ion. And if it's more, it absorbs and forms the acid. I'm going to discuss about carbohydrates and lipids. Carbohydrates, they consist of carbon, oxygen, and hydrogen. Monosaccharides. Monosaccharides are the simplest form of carbohydrates, simplest type of carbohydrates, because it contains just single sugar. Monosaccharides generally have a combination of carbon, hydrogen, and oxygen atoms in the ratio of 1:2:1. The formula for monosaccharide is CnH2nOn, where there is one molecule of carbon, 2 of hydrogen, and 1 of oxygen, and N is the number of carbon atoms. Glucose is the widely used monosaccharides. When glucose forms a ring, there are two possible arrangements of OH group, which is bound to be carbon at position 1, either alpha glucose or beta glucose. Two different forms of glucose are isomers. Isomer is a molecule that has the same chemical formula as another but different arrangement of atoms. So basically they have the same chemical formula but different arrangement of atoms. The different OH group on glucose can give chemicals different properties. Human can easily digest starch, which has alpha glucose. Cellulose has beta glucose and is completely indigestible for humans. So human can digest only starch, which has alpha glucose. Here you're gonna see the structure of ribose, fructose, some common monosaccharides. Here in this diagram, I draw the structure of alpha glucose and beta glucose. You can see the only difference in alpha and beta glucose is the presence of OH group at carbon 1. Sugars have more polar functional group attached to them. That make them very hydrophilic, which means small sugars are highly soluble in water. Monosaccharides are sweetest. Monosaccharides are sweetest. As the number of monosaccharide unit that are linked together increases, sweetness decreases. So it's inversely proportional. So we can say sweetness is inversely proportional to number of monosaccharide units. Disaccharides. Carbohydrate molecule that is made of two monosaccharides. So you can see from the term di means two. So carbohydrate molecules that is made from two monosaccharide unit joined by synthesis reaction, it's called disaccharide. Maltose is a disaccharide which forms through the linkage of alpha glucose molecules with oxygen. Linkage of alpha glucose molecule with oxygen as a bridge between 1 carbon of the glucose unit and 4 carbon of the second glucose unit. So 1 carbon of one unit and then 4 carbon of the another unit. Bonds of this type, which link monosaccharides into larger carbohydrates, are called glycosidic bonds. Disaccharide carbohydrate molecule contain same functional groups that make monosaccharide hydrophilic. Therefore, they are also very easily dissolvable in water. Here is the formation of maltose. Glucose, 2 molecules we have glucose. So this is 1, this is 4, and then the water is gonna be removed, and then it leads to formation of maltose. Complex carbohydrates, which are polysaccharides. Hundreds of thousands of monosaccharides can link together to form complex carbohydrates. Some complex carbohydrates are important for energy storage in cell, while others are essential for structural support. Now for the storage carbohydrates, examples are starch and glucose. And example of structural complex carbohydrates, examples are cellulose and chitin. These two provide the structural support. A polysaccharide molecule is a chain of monosaccharides with many subunits joined by glycosidic linkage. The dehydration synthesis reaction that assembles polysaccharides are the example of polymerization. The most common polysaccharides are plant starch, glycogen, and cellulose. These three are the most common polysaccharides. Cellulose is the main component of cell wall and most abundant organic material on earth. Cellulose is the main component of cell wall. Polysaccharides may be linear, unbranched, or they contain branches in which side chain of sugar units attached to main chain. So either they can be linear unbranched or they can be in branches. Polysaccharides are hydrophilic, but because of their huge size they are unable to dissolve in water. So they are hydrophilic. So polysaccharides are hydrophilic like monosaccharides and disaccharides, just because they are huge in size, that's why they are not able to dissolve in water. Paper towels are made of cellulose, that's why they attract water and absorb it, but they do not dissolve in water. Here is gonna be monosaccharide, disaccharide, and polysaccharides. All three are types. And structure, their structure is alpha ring, beta ring, or chain. Monosaccharide structure, polysaccharide structure, their functions and their examples. Lipids, non-polar biological molecules made of mostly carbon and hydrogen and lesser amount of oxygen. These are smaller than complex carbohydrates, so they are not considered as macromolecules because they are smaller than complex carbohydrates, that's all. Generally non-polar, so cannot dissolve in water. Usually they are non-polar. Their insolubility in water contributes their ability to form cell membrane. Some lipids are stored by cell to be used as energy source. Others serve as hormones that regulate cellular activities and act as vitamins. 5 main category: fatty acid, fats, phospholipids, steroids, and waxes. First we're going to discuss fatty acids. Structural backbone of most lipids derived from fatty acid. Fatty acid contains single hydrocarbon chain with the carboxyl function group at one end, and COH group gives fatty acid acidic property. As their chain length increases, they become less soluble. So more the length of fatty acids, they will be less soluble. If hydrocarbon chain of fatty acid binds to maximum possible number of hydrogen atoms, and if all the carbons are linked to each other with single bond, the fatty acid is called saturated fatty acid. If there is double bond, then it's unsaturated fatty acids. Fats. Fat is a lipid that is made of two molecules, fatty acid and a glycerol molecule. In a fat molecule, one to three fatty acid chains are joined by single glycerol molecule through dehydration. Synthesis between OH functional group on the glycerol and carboxyl functional group on the fatty acid. Triglycerides are most well-known fats. They contain three fatty acid chains. A fat molecule can have identical fatty acid chain or different fatty acid chains linked by glycerol. Either they can have same or they can have identical, or they can have fatty acid chains linked to glycerol. So here is glycerol dehydration synthesis, and then it forms triglyceride. Fats obtained from animals such as butter and lard usually contain saturated fatty acids, so they are called saturated fats, and they are not bad fats. Whereas unsaturated fats are good fats, and they derive from plants, contains more unsaturated and polyunsaturated fatty acids. Phospholipids. Primary lipid of cell membrane is phospholipid. Phospholipid contains two hydrophobic fatty acid at one end attached to hydrophilic polar group, which often called head group. Molecule that contains both hydrophilic and hydrophobic regions are called amphiphilipids. Here in this chart, I already explained fatty acid, fat and phospholipids in detail. Now we're just gonna look at steroids and wax. Steroids are 4 carbon rings, and their function is hormone signaling cell response to the environment and growth. And examples of steroids are testosterone and cholesterol. Wax is a long fatty acid chain linked to alcohol or carbon rings. Their function is to water resistance and protection, and examples are wax coating on fruits and leaves and stems. All proteins are polymers that are composed of amino acid monomers. So they are monomers of amino acid. An amino acid has a central carbon atom, okay, attached to the amino acid group. Here is the amino acid group, as you can see, and then a carboxyl group, okay, and then hydrogen. There are 20 different amino acid side groups or R groups, ranging from a single hydrogen atom to complex carbon chains or rings. There can be 20 different types, okay? These R groups can be 20 different types. Side groups can be polar or non-polar. Polar, which can easily dissolvable in water, non-polar, which are not able to dissolve in water, and hence they give different properties, because if the R group is different, the properties are gonna change with different R group. 8 out of 20 amino acid are considered as essential amino acid. Rest can be synthesized by cell. So 8 of the amino acid our cell cannot synthesize. Rest our cell can synthesize. Proteins. Protein can be structural, defensive, or hormones. Structural protein function is framework support. Example, hair, tendon, ligament. Defensive protein the function of defensive protein is to fight infections. For example, antibodies in our body. Signal proteins. They act as a messenger. For example, hormones. These hormones gives the message to different organs to perform their functions. Carrier proteins. Their function is to transport material like hemoglobin. So protein can be recognition and receptor, and then there are gonna be enzymes. Enzymes act as like catalyze the reaction. Examples are amylase, and then motile. The function is movement. Obviously, motile we can say movement. And then examples are actin and myosin, which is present in our muscle for the movement. Protein structure. Proteins have 4 levels of structure: primary structure, secondary structure, tertiary structure, and quaternary structure. Primary structure is linear structure of amino acid in each peptide chain. Single change can alter or even destroy biological function of protein. Secondary structure. Most peptides have proteins that are rapidly coil or fold into patterns and contribute to the overall shape of the protein. It is result of hydrogen bonding between different parts of the same amino acid backbone formed by hydrogen bonding. Okay. 2 common structures are beta plated sheets. Example is like they are found in strands of silk, or alpha helix found in filament protein and transmembrane protein. Tertiary structure. Overall 3-dimensional structure or shape of a protein due to range of bonding interactions among the amino acid. The intermolecular reaction of the R group determine the 3-dimensional shape of the polypeptide chain. So R group, intermolecular reactions of R group will determine the shape of the polypeptide chain. These intermolecular reactions include ionic bonds, hydrogen bonds, hydrophobic interactions, and disulfide bridge. Tertiary structure is important to functions of protein, especially enzymes. Extreme conditions can unfold a protein, causing denaturation, a loss of both structure and function of protein. So denaturation is loss of structure as well as function. Denaturation can be caused by rising in temperature, can lead to denaturation of protein. Now we are going to study about quaternary structure. Many proteins are composed of 2 or more polypeptides that come together to form the final functional protein. That is called quaternary structure. Item in our book describe all the 4 structures of protein. This is primary, then secondary, the beta plated or alpha helix, and then it forms tertiary structure. And here you can see hydrophobic interactions, hydrogen bonds, disulfide bridges, and ionic bonds in the tertiary structure. Nucleic acids serve as assembly instruction for all the proteins in living organism. There are 2 types of nucleic acid, either DNA or RNA. DNA, each nucleotide contains deoxyribose, a phosphate group, and one of the 4 bases, which can be either adenine, thymine, guanine, or cytosine. Whereas in RNA, each chain nucleotide contains ribose, a phosphate group, and one of the 4 bases, adenine, uracil, guanine, or cytosine. The only difference is in RNA it's uracil, and then DNA it's thymine. Nucleotides perform many functions in the cell in addition to serving as building block of the nucleic acid. ATP and GTP are primary molecules that transport chemical energy from one reaction to another. These two are the molecules which are responsible for transporting of energy. Enzymes are catalysts, usually a protein that speeds up a chemical reaction. Enzyme binds to a specific reactant called substrate. Enzyme specificity explains why a typical cell needs about 4,000 different enzymes to function properly, because a specific enzyme can join to a specific substrate. That's why for different substrates we need different enzymes, right? Active site. What is an active site? It's a pocket formed when newly synthesized enzyme fold into its correct three-dimensional shape. Enzyme substrate interaction can be explained by lock and key hypothesis. So enzyme is a lock and it can be opened by a specific key. So here key is gonna be substrate and lock is enzyme. Induced fit hypothesis. Before binding, an enzyme changes its shape so that the active site becomes more precise in its ability to bind its substrate. This is called induced fit hypothesis. This is an enzyme. Here is an active site. It will become more precise so that the substrate can attach here. This is called induced fit hypothesis. So now what are cofactors and coenzymes? Cofactors are non-protein group. Cofactors are non-protein group that binds very precisely to an enzyme. They are often metals such as iron, copper, and zinc. They are absolutely essential to for the catalytic activity of an enzyme. What are coenzymes? They are organic cofactors. So basically the cofactors, but they are organic, play a similar role as cofactor and are often derived from water-soluble vitamins. Nicotinamide adenine dinucleotide NAD positive derived from B3. This is an important coenzyme and it acts as an electron carrier during number of biochemical pathways. Now we're going to discuss about factors and conditions that affect enzyme activity, enzyme substrate concentration, temperature, and pH. First, we're going to discuss about enzyme substrate concentration. If excess substrate present, if the substrate is in excess amount, rate of reaction is proportional to the enzyme concentration, because the amount of enzyme limits the rate of reaction. If the substrate is in excess, the rate will be directly proportional to enzyme concentration. If the amount of enzyme is constant, then if the substrate increases, rate of reaction increases to a point called saturation level. Here are the graphs. It says first it increases as the substrate increases and then it reaches to a point which is called saturation level. Enzyme inhibitors lowers the rate at which an enzyme catalyzes the reaction. Inhibitors work by binding to active site of an enzyme. Some inhibitors bind to critical sites located elsewhere in the structure of an enzyme. Inhibitors have shape resemblance to normal substrate closely. So inhibitors resemble to the substrate very closely. So we are going to study two types of inhibitions here. One is competitive and one is non-competitive. In competitive inhibitors, the inhibitor actually competes with the normal substrate for access to the active site. Inhibitor attached to the active site, so basically it gives competition to substrate. In non-competitive inhibitor, they inhibit activity of an enzyme, but they bind to a location other than an active site. This changes the shape of the enzyme so that the substrate can bind efficiently. So let's say if this is an enzyme and this is the active site, right? And this is a substrate. and this is inhibitor. So this is a substrate and this is inhibitor. So they are all almost same in shape, and they compete with each other to bind to the active site. Whereas in non-competitive inhibition, the inhibitor is gonna bind to somewhere else, let's say here, and it will change the shape. So if this is the shape of the active site where the substrate is gonna fit, they are gonna make it from this here this fits. So with the inhibitor attached, it's gonna be like this, so that the substrate can't attach the active site. This is called non-competition inhibition. Now, allosteric control of an enzyme. Molecules that naturally regulate enzyme by binding to its active site, called allosteric site, and cause a change in the shape of enzyme and thus affect the active site and thus affect the active site. This type of regulation, in which proteins function at on site is affected by a molecule binding to a separate site, is called allosteric regulation. Allosteric regulation may either inhibit or stimulate enzyme activity. It can either inhibit or stimulate. If the product accumulates in excess, its effect as an inhibitor automatically slows down the or stops the enzymatic reaction. If the product is available in excess now, it starts acting as inhibitor, and then it slows down the enzymatic reaction. And if the product is less, the effect of the inhibition is reduced and the rate of reaction is increased. Regulation of this type, in which product of a reaction act as a regulator, is called feedback inhibition. So the product, if the product is less, it will increase the rate. If the product is more, it will decrease the rate. So if the product is controlling the rate of reaction, is called feedback inhibition. Feedback inhibition is important because it prevents cellular resources from being wasted. Here is an example. Enzyme, this is the allosteric site and this is an allosteric activator. This is active site and this is substrate. When enzyme binds to an allosteric site, okay, when this enzyme binds here, the enzyme will be in now highly affinity state, and the substrate can bind easily. This is called allosteric activation. On the other hand, when allosteric inhibitor binds to enzyme, binding of inhibitor converts enzyme to low affinity state, and then the substrate will be released. The example of allosteric inhibition is the formation of threonine from the amino acid leucine. So when threonine is in excess, it will slow down the process, and when the threonine is less amount, it will increase the process of the formation of threonine. pH and temperature effect on enzyme. Enzyme has an optimal pH where it can operate at its highest efficiency. The pH either increase or decrease from its optimum value. The rate of catalyzed reaction will be decreased. More the pH value deviates, more extreme are the effects, until the reaction rate falls to 0. Most of the enzyme function at pH 7, but pepsin is an enzyme which is works in acidic conditions found in our stomach. Protein digesting enzyme, it works efficiently in pH of 1.5, whereas trypsins work in alkaline pH of 8. The effect of temperature changes on enzyme activity reflects two distinct processes. First, if the temperature increase, rate of reaction increase. From 0 to 40 degrees Celsius, rate of reaction doubles for every 10 degrees change in the temperature. Above 40 degree, increasing kinetic energy motion denature the structure of an enzyme, and enzyme will lose the ability to function. But again, there are a couple of exceptions. So enzymes of corn pollen function best near 30 degrees Celsius, whereas the enzyme found in Antarctica fish, they work best at 0 degrees Celsius. And single cell archaea function, the enzyme of single cell archaea function, because they found in a very high temperature region, so they function best at 85 degrees Celsius or above. Application of enzyme plays a very important role in animal feed, brewing, dairy, detergent, leather, starch, wine and juice industries.